Hello, everyone, and welcome to Ocean Acidification Week 2020, a virtual multi-day forum to highlight ocean acidification research and initiatives. This is the first session of Ocean Acidification Week. And OA Week is presented by GOA-ON, the Global Ocean Acidification Observing Network, NOAA, the National Ocean and Atmospheric Administration, the IAEA, the International Atomic Energy Agency, Ocean Acidification International Coordination Center, and the IOC-UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. This session will feature talks from the Pacific Islands and Territories. We are so glad to have over 140 registrants for today's session, who are joining us from all over the world. My name is Michael Aquafreda, and I work with the GOA-ON Secretariat. I'm also a Knauss Fellow working with the NOAA Ocean Acidification Program, and I'll be moderating today's session. During the presentation, all participants are in listen-only mode. You are welcome to type any questions into the question box, which can be found at the bottom of the control panel on the right-hand side of your screen. You could also post questions on Twitter using the hashtag OAWeek2020. We'll be moderating incoming questions, and we'll pose them to our speakers during the question and answer section, which will begin immediately after the last presentation. Today's webinar is being recorded and will be available through the GOA-ON YouTube channel soon after the OA Week ends. Before we hear from our speakers, I would like to share a short presentation about GOA-ON. GOA-ON is a collaborative international network designed to address three high-level goals: improve our understanding of global ocean acidification conditions, improve our understanding of ecosystem response to OA, and acquire and exchange data and knowledge necessary to optimize modeling for OA and its impacts. GOA-ON offers its members a number of resources, including informing policy, mentorship, providing data, and also hosting webinars. OA Week is the first set of GOA-ON webinars, and GOA-ON webinar series will continue every 4 to 6 weeks for the time being. The data portal offers a chance for scientists around the world to access and visualize ocean acidification data, which is being collected from a wide range of sources, including moorings, research cruises, and fixed-time series stations. GOA-ON mentoring occurs through a mentorship program called the Peer-to-Peer Network. And this network matches senior researchers with early career scientists to facilitate an exchange of expertise and provide a platform for international collaborations. Scholarships and trainings are also available through this mentorship program. And at the international level, GOA-ON informs policy. GOA-ON works with the UN to develop the SDG 14.3, or the Sustainability Development Goal 14, which is about life below water. GOA-ON members assisted with the development of the indicator methodology, which provides guidance on how to measure ocean acidity and how to report that collected information. GOA-ON is also involved with the Commonwealth Blue Charter, and it is also involved with the UN Decade of Ocean Science for Sustainable Development, 2021 to 2030. As of 2020, GOA-ON has over 750 members from over 100 countries. There are 8 regional hubs, and we'll be hearing from most of those hubs throughout Ocean Acidification Week. You could join GOA-ON at any time by logging on to the GOA-ON website or using this link here. Initially, the 5th International Symposium on the Ocean in a High CO2 World was scheduled to take place this week in Lima, Peru. Yet, like many other events scheduled for this year, the symposium has been postponed until 2021 because of the COVID-19 pandemic. This symposium would have brought together researchers, decision makers, and other stakeholders to share cutting-edge OA science. We're looking forward to that meeting and hope everyone listening today will visit the symposium website and learn more. We'll hopefully see you in Peru in 12 months. Today, we'll be hearing from 6 different researchers, and they include Duncan McIntosh at SPREP in Samoa, Kim Currie in New Zealand, Antoine from the University of South Pacific in Fiji, Krishna Kotra from the University of the South Pacific in Vanuatu, Patilla Malua Amosa from the National University of Samoa, and Manuba Rota from the Ministry of Fisheries and Marine Resource Development in Kiribati. Before we get started, I'd like to talk a little bit about Pacific Islands and Territories, or the PI-TOA Hub. The term TOA means warrior in many Polynesian island languages. The PI-TOA network works collectively to better understand and combat the impacts of ocean acidification in the region. In recent years, there have been several ocean acidification trainings and GOA-ON in a box kit recipients in the Pacific Islands and Territories. As capacity for ocean acidification monitoring increases in the region, there's an increasing need for collaboration and communication among the various islands and territories. And since its founding, there are now 28 members of the PI-TOA Hub, and they come from 12 different nations. So without further ado, I'm going to pass it over to our first speaker, Duncan.
Thanks, Mike. Can you see my screen okay?
Yes. You'll just want to make it in the full screen mode. That looks great. Take it away.
Okay, great. So I'm just going to share briefly about a project that SPREP is implementing in the Pacific. It's called the Pacific Partnership on Ocean Acidification. So first, for those in the audience who may not be familiar with SPREP, we are the Secretariat of the Pacific Regional Environment Program, and we are an intergovernmental organization that was established for the protection of the Pacific Islands environment. And our headquarters is in Samoa, where I'm coming to you now. And our members include 21 Pacific Island countries and territories, as well as 5 metropolitan members made up of the larger countries, Australia, France, New Zealand, UK, and the USA. Some background on this project: the Pacific Partnership on Ocean Acidification stemmed from side conversations held during the 3rd UN SIDS conference that was held in Apia, Samoa in 2014. And these conversations identified a need in the Pacific region for monitoring ocean acidification, for building capacity and resilience, and for implementing adaptation strategies to ocean acidification. So this project is run as a partnership, and it's a collaborative effort between SPREP, the University of the South Pacific, the Secretariat of the Pacific Community, and this project is financially supported by the New Zealand Ministry of Foreign Affairs and Trade and by the government of the Principality of Monaco. The project was designed with 4 focus areas, and those are research and monitoring, capacity building, policy support, and adaptation actions. For the adaptation actions, we have established 3 pilot sites, and one is in Fiji. We have pilot sites in Kiribati and pilot sites in Tokelau. On the research focus area, to date, we have published a regional OA vulnerability assessment for the Pacific Islands, and that's available at this link, and you can find it on SPREP's website. We've also published a vulnerability assessment of pelagic fisheries for the Pacific. And we also have established, in partnership with the University of Newcastle, Australia, a PhD scholarship for Pacific Island students. And to date, we have a student from Papua New Guinea who is now onboarding at the University of Newcastle, and she is starting her research in partnership with our pilot sites, where she'll be investigating the effects of coral restoration on coral reef resilience to ocean acidification. On the awareness focus area, we have published a number of different outreach materials, and one example would be these ocean acidification informational posters, which we have translated into 7 Pacific Island languages so far and distributed to different ministries and schools across the Pacific. On the monitoring focus area, we have a great partnership with GOA-ON and the Ocean Foundation, where we have provided equipment and training to 8 Pacific Island nations so far. And these are in the form of what's known as a GOA-ON in a box kit. And it's basically all-in-one kit that includes the spectrophotometer, the instrument you see on the bottom right, and it includes monitoring equipment to take water samples and investigate total alkalinity so that these Pacific Island nations can report data against the Sustainable Development Goal indicator 14.3.1, which is marine acidity, pH, to be measured at representative sampling stations. Also on the monitoring focus area, we've partnered with the Korean Institute of Ocean Science and Technology in a project that is installing MAP CO2 OA monitoring buoys. And to date, we've installed one in Palau, one in Federated States of Micronesia on the island of Chuuk, and one is here in Samoa, soon to be installed, currently delayed by COVID-19 travel restrictions. On the adaptation focus area, we've held numerous iterative rounds of stakeholder consultations so that this is a bottom-up effort that's driven by stakeholder priorities to determine what types of adaptation actions the communities want to take in response to ocean acidification. Some of the options that are on the table for these communities include enhancing primary producers to locally buffer pH, and that can come in the form of seagrass or mangrove restoration, because these plants, these aquatic plants, as they photosynthesize, can take up some of that CO2 that would otherwise lead to ocean acidification. And this can have a local buffering effect for any calcifiers that are in the nearby vicinity, like coral reefs. There's the adaptation option of coral restoration, which can enhance reef resilience, either by selecting a specific species that may be more resilient, or by enhancing the overall ground cover or species diversity of coral on a particular reef. Adaptation options also include reducing other reef stressors through management practices. So that may look like establishing locally managed marine areas or marine protected areas. That can include conditional closures, rotational closures, permanent closures. In the Pacific, this is not a new idea, as traditional management practices for millennia have included different types of marine closures. In Fiji, this is a tambutara, or a rotational closure where certain species may be off limits for a spawning period, for example. Another adaptation option to reduce pressure on the reef can be directly reducing fishing pressure by providing alternative livelihood for reef-dependent communities. For example, supporting aquaculture in a community that may be dependent on nearshore reef fisheries for food security. In Fiji, the pilot site that we have established is on the island of Taveuni, and the stakeholders met and determined that their priorities for adaptation will include mangrove restoration, the development of locally managed marine areas, establishing alternative livelihood, and our partners in Fiji include the Fiji Locally Managed Marine Area Network, FLEMA, Conservation International in Fiji, and the Wakatu Foundation in Fiji, which does great educational outreach. In Tokelau, the stakeholders wanted to focus on coral reef restoration. So we have started to establish coral reef restoration on both the lagoon side and the ocean sides of all 3 of the Tokelauan atolls, and those are Atafu, Nukunonu, and Whakaofo. And in Kiribati, you will hear more specifically about our work in Kiribati from our partner Manibua, but we have worked in partnership with the Ministry of Fisheries there to establish a locally managed marine area in the village of Nanikai on the atoll of Tarawa. And this LMMA includes seagrass restoration, clam restocking, mangrove restoration, and coral restoration. So it's a comprehensive LMMA there. On the policy support focus area, we have held specific trainings for Pacific Island policymakers on developing policy options to address OA, and we have published this handbook for Pacific Islands focused on mainstreaming ocean acidification into national policies. So this handbook is available on SPREP's website as well. Next steps for the PPOA project. We are currently seeking additional support, and we have some commitments so far from Monaco to extend and upscale this project. We have interest from quite a few of our SPREP members who did not yet establish pilot sites for adaptation to expand this type of work to other SPREP member countries. And happy to take questions during the question and answer session, and I'll hand off to our next presenter by saying Faftailava. Thank you very much.
Thank you so much, Duncan. I'm going to switch the presenter over to our next speaker, Kim Currie.
Kia ora everyone, can you hear me okay and see my screen?
Yes, we can, Kim. We could hear you and see you.
Kia ora koutou, talofa, bula, ko ngā Māori. Good afternoon everyone, it's Kim here from NIWA in New Zealand. It's great to be sharing science with you, ocean acidification science, seeing some of you who I haven't seen for quite a long time, and hearing what you've been up to in the ocean acidification field for the last couple of years. I'd like to talk today about a new project, collaborative project that we have going in New Zealand between the New Zealand Ocean Acidification Observing Network and the Department of Conservation. This is a joint presentation between Monique Ladd from the Department of Conservation, Te Papa Atawhai in New Zealand, and myself, Kim Currie. I work for the National Institute of Water and Atmospheric Research in New Zealand, and I'm responsible for the New Zealand Ocean Acidification Observing Network. Unfortunately, Monique's not able to join us today, so I'll be presenting the whole thing with input from Monique. So the New Zealand Ocean Acidification Observing Network provides an observational framework for working with ocean acidification from a chemistry viewpoint. So the network consists of 10 core sites across diverse ecosystem types. It includes the UNITA time series, a 22-year-old time series that collects climate quality data, and several short-term sites. The network provides a baseline of data of present-day conditions and variability on the monthly, seasonal, and interannual variability. And this is providing a baseline against which we hope to measure future change, given that the program will extend into the future. It allows us to address regionally specific scientific questions, and we also contribute to GOA-ON, both from the data set point of view, but also contributing and participating in events like this. Sampling partners at each of the sites are provided with self-contained sampling crates, like the ones shown in the picture on the bottom. The sampling partners take the samples, then return the crates to a central laboratory in Dunedin, New Zealand, where I'm based, and where we measure DIC and alkalinity, and that allows us to calculate the other parameters. The data is available on our NZOA-ON website, the data and some graphics available to everyone, and we also provide it directly to our sampling partners after each batch of analyses. We're linked to the GOA-ON portal. That picture on the bottom shows a close-up of the New Zealand portion of the GOA-ON portal, where you can see our sampling sites with the appropriate metadata and the link to the website with the data itself. The data from the New Zealand Ocean Acidification Observing Network was submitted to the United Nations as part of New Zealand's reporting to the Sustainable Development Goal 14.3. It's also been used by the Ministry for the Environment in the Our Marine Environment report and was featured in the WNO Provisional Statement on the State of the Global Climate in 2019. The data is able to inform experiments, perturbation experiments and others, and has formed the basis for several university and school projects looking at time series and variability of ocean acidification. And there's also scientific publications. Our sampling partners at the different sites around the country are from a variety of institutes, including local government, research institutes, the aquaculture industry, the fishing industry, iwi, a port authority, and the Department of Conservation is our sampling partner at a marine reserve in Stewart Island to the south of New Zealand. From the data collected in the 5 years we've been running so far, we've been able to determine monthly mean conditions and fit seasonal trends to the data from each site. The diverse ecosystems covered by the sampling sites are represented by different drivers which we've been able to start to tease out. However, New Zealand has a really long coastline and covers a large latitudinal range and a diverse range of marine environments. So that has resulted in a lot of areas that are undersampled, where we don't have much idea of what's going on with ocean acidification and carbonate chemistry. The Department of Conservation, Te Papa Arewhai, is committed to monitoring climate change, both in the terrestrial environment and in the marine environment. They look after a suite of marine reserves, which, among other things, provide dedicated areas for scientific research. DOC have committed to implement ocean acidification monitoring in 9 of these marine reserves, and they are working with the treaty partner and other local communities to collect water samples. The Department of Conservation, Te Papa Arewhai, wants to understand the impacts of climate change at a national scale and have chosen indicators of pH and water temperature and are planning monthly sampling in the marine reserves. Currently, the sampling is mostly done at the local level, and only 1 marine reserve is currently monitored for pH. That's the one I showed you in the south of New Zealand on Stewart Island, Rakiura. However, national scale monitoring at the 9 marine reserves will give insight into natural variation and vulnerability of the different bioregions. So the ocean acidification sampling started last week, and Dr. Monique Lads from DOC collected the first sample in the Tuhua Marine Reserve. So that was exciting to start the implementation of the Marine Reserve monitoring and to have that in conjunction with OA Week. So that's Monique taking the first samples with the sampling crates. So with the advent of the 9 marine reserve sampling sites joining into the network, that has really increased the coverage of OA monitoring around New Zealand. And so the collaboration between the Department of Conservation, Te Papa Arewhai, and NIWA and the New Zealand Ocean Acidification Observing Network, we're looking forward to a great future with that. The collaboration is not just between the institutes, but it's between people. And it's been great working with Monique from the Department of Conservation as we get this project underway, and look forward to several years, many years hopefully, of continued monitoring. So I'd like to thank particularly Judith Murdoch, from the University of Otago, who manages the logistics and the lab associated with the New Zealand Ocean Acidification Network, and our sampling partners throughout Aotearoa New Zealand, including our new DOC sampling partners. Thanks very much, and I look forward to being part of the discussion at the end of this session.
Thank you so much, Kim. And yes, as a reminder, all of the questions will be answered at the end of all of the presentations. But if you have a question at any time, please put it into the question box on your dashboard. All right, our next speaker is Antoine, and I will make him the presenter right now. Antoine, I just made you the presenter.
Good afternoon, good evening. This is Antoine from Fiji. So today we'll be discussing about monitoring of ocean acidification and anthropic impacts on the Suva Lagoon and Reef. And for long-term monitoring of Suva Lagoon and the area in Fiji. So through the generosity of Ocean Foundation, we do have GOA-ON pH kit that was donated in 2017 and which has been shared among staff here. Also, we have a National Oceanography Center of the UK collaboration with a prototype pH sensor and nitrate sensors, and these have been deployed also. The other equipment which we have is a RBR dual salinity sensor and a CFET pH sensor. Through the first GOA-ON IFSAMI deployment, we had some pH variability studies done in Suva Lagoon over a 24-hour period. And we did detect some very high diurnal biological variability in pH. And discrete water sampling that was done also matched very well with those readings from the ISAMIs, which were a good thing. So you can see that there's a very high variability in the pH that was due to biological activity. Later, we did deploy another pH sensor, which is a spectrophotometric based sensor with microfluidic technology, which is quite precise. And the first deployment, which was about a month, produced the time series that you can see now. And you can still see the variability here in the biological activity. But to get the climate change signals, of course, we'll have to try and characterize this biological signal and remove it from the baseline climate data. And here on the photo, you can see the frame holding the pH sensor as well as another equipment there, which is measuring the oxygen as well as temperature. And also we have a salinity sensor. This is some pictures of the first deployment with some of the postgraduate students, where we used underwater parachute to deploy the sensor frame. Here is a very short video showing how it was deployed. So you can see water is very murky, and also it's not, you can see some effects maybe of turbidity. There are not too many live corals in the area. It would be interesting to see the data from the sensor on the long term. We did have real-time telemetry on this sensor, which was since removed because of theft and other issues. So this is some of the data that came back, and you can see that DO and pH are very closely related. And that does indicate that there's a lot of biological activity going on there. And as well, you can see the salinity here as well as the temperature. The next phase, which will start in 2021, is a project on anthropic impacts on net productivity in the Suva Reef. This is in collaboration with the IRD in New Caledonia, the National Oceanographic Center, and involves USP. So here we'll be looking at the four things, mostly anthropogenic, the river input, climate change, on the biogeochemistry, the ocean currents, and also biological species. So ocean acidification will fall on the biogeochemistry aspect, where we'll be monitoring ocean acidification in the lagoon and outside of the reef. So we'll deploy also this nitrate sensor, which will be very useful to look at the impact of the river from sewage and other inputs. As well, we'll measure high-resolution scale ocean acidification data. We're hoping to get perhaps a PhD student or a master's student to work on this, with one of the main aim being to see if the Suva lagoon is net respiring or net photosynthesizing to input into this production respiration model. And also we'll be carrying out TA sampling on a regular basis to determine the calcification levels, as well as monitoring the effect of sedimentation on net productivity. So it's to get overall idea of how river input and ocean acidification will impact fisheries production in the lagoon, and of course photosynthesis, and how much is it contributing to a carbon dioxide intake, or is it negative or positive? So that's all we have today, and I'll be welcome to take questions later on. Thank you very much.
Thank you so much for that, Antoine. Next up, we have Krishna. Krishna, you'll need to unmute yourself, but we could see your screen great.
Are you able to see my screen? No?
Yes, we can see your screen and we could hear you. You're good to go.
Okay. Thank you. Good afternoon to everyone from our time. Today I would like to give a brief on the ocean acidification being monitored in Vanuatu. We started this ocean acidification monitoring in Vanuatu in 2018, where we held a week-long workshop with different participants from the government ministries, who are the policymakers, and so many private organizations, community leaders, and apart from the students at the USP were involved in this week-long program. It is also the part of peer-to-peer network program, where the mentor came and we had a great workshop. And it was a first of a kind in Vanuatu, where ocean acidification being explained to these communities and policymakers, and we could really present that there is a need to have this to be monitored in a high level with the help of the government. And we thought that we really successfully made this to be presented to the policymakers at that week-long presentation. Then after the workshop conducted, we deployed our ISAMI that was being donated by the Ocean Foundation. We had this in the capital, where we chose the site called the Aotearoa Lagoon. You can see in the map as well, with its latitude and longitude. And we were mainly concentrating on taking the pH and alkalinity besides salinity and temperature. And initially we thought that it would be a bi-weekly sample collection and analysis, and later it was shifted to 3 weeks sample collection. Could see in the picture below the site and the ISAMI that is being in the shore after being retrieval. And as part of the continuous programs, we have been making sure that students are being part of this monitoring program. These are some of the pictures that were taken during the training and deployment, where you could see some community leaders are also part along with the students, where we have make sure that not only the USP, the campus is involved in this ocean acidification, but it is widely published in the public domain, and the ocean acidification topic should be discussed among the students into their communities, and they can further explain what is being happening in and around their seas and how different ecological systems and the fishing community being impacted by this ocean acidification. Soon after the deployment and the retrieval, we started to face so many problems, especially the troubleshooting of the ISAMI, where we have to completely open it up and take discussions with the service provider, and then we completely reset it so that it would work normally. So it was a period where we have to really stretch ourselves through the email communications and follow the directions, had some calls, all these things. Then again, after fixing the ISAMI, again we have problems with fixing with our multimeter, where there are some problems with the adapter changes, voltage fluctuations, and the readings are not going well. So we have to fix that as well. That also took us a quite bit of time, because here in this Port Vila, we couldn't really know who could really fix this multimeter. Then once we fixed the ISAMI and again deployed in the site, again we could see some more problems in regards to the malfunctioning of the ISAMI software. Even though the sample was injected into the water, but we couldn't really get the pH values. In the screenshots you can see, even though you could see the temperature and salinity being given it with the batteries and battery voltage, but you could see the pH is being constantly shown as 0. So this was another problem that we encountered and were stalled for some quiet months, and we can again, after a few deliberations, discussions, instructions, and all these kinds of things, again we could slowly start the work back again. In meantime, we had collected some small data for the pH. In the screen you could see around 1,000 readings we have collected, and here you could see some fluctuations here and there in the pH range in the lagoon. We have not finalized any conclusions with the short data, but once we have a comprehensive data for over series of time, then we will really see how these fluctuations in pH mean. Meanwhile, we also collected some salinity data as well with the temperatures and pressures, specific conductivities as well. So we will incorporate this into the pH and see how both being varied with the time series. That also do when we have good comprehensive data that is being collected. As far as this small period of installation, we could see there is a huge success both in the introduction and awareness to the ministries and NGOs, communities, and students are being part of this project. And so far, for several months, we have been collecting pH, alkalinity, temperature, and salinity of the lagoon, and we are storing the data. And we think that because of some of the issues that raised during the operation of this data collection, but we are looking forward that we are slowly progressing and steadily progressing, and with the small expertise and experience that now we got in ocean acidification. In coming days, we would be really feeling comfortable in taking this program at a larger level, like increasing the number of sites and making sure some more funding will be coming where students can be employed or part-timers will be employed to have a continuous data assessment. So we think that we are really slowly moving towards to a better position from where since the last 1.5 years. During this course of time, I could really see there are some challenges in regards to there is an expertise that because I myself is doing alone here. So sometimes if there is any troubleshoot, they really has to really reach to the other sources and take feedback and do accordingly. So, but slowly, as I told you earlier, that we are reaching there now with small experience that we got. And there is another challenge that is also in regards to the time, because I am a full-time teaching faculty here and myself has to go to the sample collection, analysis and all these things. But so far we are managing with the help of other staff who are helping, and the students are also helping in the sample collections and analysis. So things are going on slowly. And the other challenges, as we have been discussing, that there is a need for space. Every time if there is something faulty being observed in the malfunctioning of any equipment or any part, there is no option except to reach to Australia and New Zealand, because in country we don't find this kind of equipment or space being supplied by any agencies or industries. So that is another issue that is a challenge for us. And at times I couldn't myself make sure that how I could manage the troubleshoot. So we have to leave some things like that without any attendance and all these things. So at current status, it's a not good status for us because we are not using ISAMI anymore because it's completely out of fixing things. So we are not sure whether we have to send it over to US to check it or how long it will take. And with the COVID pandemic, we are not sure of the dates and all those things. And also there is an electrode for the alkalinity, which is again giving us from cranky values. So we are also awaiting for an electrode, which we ordered from New Zealand. And again, there is some disturbances in the working of the spectrophotometer as well that we are discussing with the company, and hope sure that we will make sure this is resolved in time and we start working again back as well. So with this introduction and the small amount of experience and over the year and a half, I could really see that there is a need for an establishment of a regional hub where we can facilitate for the Pacific Island countries to have some space or instruments or some exchange of equipment on a time basis where someone having problems. And also this regional hub to me would able to have some conversations in regards to troubleshoots and discussions. Yes, there are many other open sites where you can post your discussion, but within the Pacific and where we can have a telephonic calls as well, talk for some at length rather than writing in emails. So I think this regional hub would be an avenue that would be a place where the researchers can resort to resolve the issues. And also it would be a place where we, especially the Pacific Islanders, will have an yearly meeting or a workshop or some time to gather to exchange our updates and see what's happening with others and any funding opportunities or troubleshoots being managed in a different way or doing the work in a different way. So all these kinds of exchanges can be facilitated at this regional hub meeting. I think it would be a great avenue for Pacific Island researchers, because we are in a place where it is always we have constraints in both the expertise and getting the equipment. So this regional hub to me will be a good place where everything will be facilitated with a quick exchange between the countries, so that the work can be running down rather than halted for how many months getting to the Europe or USA side to get the equipment for us to get started back again. So we kindly acknowledge the funding opportunity given by the Ocean Foundation. They are so cooperative since start of this monitoring program, and we hope and continue that their assistance will be continued in the future as well. Thank you, Thomas.
Thank you very much, Krishna. Our next speaker is Patilla.
Yeah. Hi, it says slightly again too. Okay, talofa and greetings from the National University of Samoa. Before I look at the challenges of our project, I'd like to talk first about an overview of our work, our measurements, and our progress to date. So this slide provides a historical overview of our partnership with the Ocean Foundation. Our MOU with the foundation was signed in 2018. COF provided the GOA-ON in a box kit, which NUS would later use to implement a project to collect OA data. We received the kit in the latter half of 2018, then conducted a training on the use of the kit materials and instruments. We started trial measurements to identify and explore potential sites for our measurements and to familiarize ourselves with the kits and instruments to be used. So our proper research measurements started in January 2019. So we used it to conduct a proper research after gaining ethics approval from the university and consent from the local community to deploy our ISAMI at the MPA. So generally, the research question was, what is the variability of carbon chemistry at the marine protected area of Savaya Village, Upolu? And based on that research question, we proposed 3 major goals for the study: to characterize the chemistry of seawater at the MPA, measure the monthly variability of carbon seawater chemistry, and to compare seasonal variations in seawater chemistry within the MPA. And to achieve those major goals, we established 3 specific objectives to guide our research. These were to measure the incident pH and temperature of seawater on a monthly basis from January 2019 to October 2020, undertake monthly lab measurements of seawater alkalinity and pH, and computes the concentration of dissolved inorganic carbon and PCO2 in seawater from our pH and alkalinity lab measurements. Just some background on Samoa. Samoa is located in the South Pacific and consists of 2 main islands, the big Savai'i Island and the smaller Upolu Island. So our village, Savaya, is located on the south coast of Upolu Island. The village is roughly 35 kilometers from the main town area of Apia, with an estimated population of roughly 400 people. And the MPA is about 52,500 square meters. So this MPA was selected based on its present coral reef coverage, accessibility, existing data related to coral and fish resources, history of successful projects at the MPA, and very importantly, security for safeguarding our project equipment, specifically the ISAMI. In addition, this project presents a great opportunity for collaboration between our university and the village community to strengthen community and publicity outreach programs. So this lower image shows an outline of the MPA, the location of our ISAMI, and the sites where we collected samples for our lab measurements. So we were able to conduct our lab measurements from January 2019 to February 2020. We did not go as planned due to challenges that I will talk about later. We undertook ISAMI measurements from January to November 2019, so currently is having problems, so we're not able to conduct further measurements at this stage. And we are currently processing measurements and analyzing data for trends and to answer our research questions. I'd just like to show some data from the ISAMI. So this chart shows pH temperature measurements from the 7th of February 2019 to 7th of March last year. So we see lower temperatures which correspond to early morning and late evening, and spikes in temperature in the afternoons. And we see pH following temperature trends, except for this part down here where there was a big drop in temperature, but thereafter temperatures seem to lag behind pH compared to these sections. So I'm not sure if it was taking out ISAMI to download data followed by redeployment. But looking at rainfall data during this period, there was no rainfall recorded at the station near this location. So this slide summarizes some of the challenges we faced from the start of the project. Some of these challenges were addressed but kept repeating, and we've also heard about some of them from the previous speaker. So we faced technical problems with the ISAMI and multimeter, as well as software update issues at the initial stages of measurements. Unfortunately, we lack expertise on island, so we had to learn on fixing problems as they arose through communicating with the Ocean Foundation from a distance and with the assistance of Dr. Duncan McIntosh from SPREP. At times, the tides were unpredictable, and the location for our OA work is quite far from work, about 2 hours travel to and back. However, this site was recommended to us by the Fisheries Division of the Ministry of Agriculture and Fisheries as we explored potential sites. We have a high staff turnover, so last year we had 2 experienced senior staff taking off for doctoral studies overseas and 1 resigning for new employment. Our current team is actually dominated by females, so we had to seek a male colleague to accompany us to assist with ISAMI deployments. And the 5th challenge now is that the restrictions of the measles epidemic last year and the university shut down due to our state of emergency restrictions prevented any data collection during those periods, as we were not allowed to work on campus in the labs. So what is the way forward for our team? We plan to continue with more OA projects by moving our ISAMI once fixed to other locations around Upolu Island, and hoping to collaborate with the relevant government ministries and non-government organizations. We also plan to continue our collaboration and partnership with other local communities. We're very grateful for the support of the Ocean Foundation and assisting us from a distance when problems arose. We were visited by NOAA and Alicia in 2019, and they provided some capacity building workshops, especially training to new members of our team, as well as assisting in technical problems that we had at the time. I'd like to acknowledge our colleagues who are participating and assisting with this project from my team here at the university and regional help through Duncan of SPREP, the Ocean Foundation, without whom we would not have the opportunity to embark on this exciting project, and of course the National University of Samoa. With that, I thank you for your attention.
Thank you so much, Patilla. All right, now we're going to hear from our last presenter, Manibua.
Hello, everyone. Can you hear me?
Yes, we could hear you and we could see your slide.
Okay. Okay. Mario from the island of Kiribati. And I am Manibu, and present with me is Mr. Max Peter, our own principal fisheries officer. And my presentation today will be based on adaptive and restoration approaches for resilient Kiribati toward ocean acidification. So there are only the outline of my presentation will focus on these 3 points, which is to give a brief background on Kiribati, then to discuss the current adaptive and restoration approaches that have been done on the island. And lastly, I will discuss our vision toward these approaches. But before we move on, I'm thankful for those previous presentations. And we now know where we are in ocean acidification project in Kiribati. So the Republic of Kiribati, as most of you know, Kiribati Island are mostly low coral atolls surrounded by extensive reefs with 33 scattered islands, and 21 of these islands are inhabited with a total population of 112,407 as of a 2015 census. Okay. Moving on to our next point, therefore, current adaptive restoration approaches for resilient Kiribati toward ocean acidification that have been done by coastal fisheries, which I will discuss in my next slide. The first adaptive and restoration approach is to establish locally managed marine area through community-based fisheries management. The main goal of this approach is to reduce reef stress and to support reef resilience. Locally managed marine areas have been successfully established in Kiribati, most island Kiribati at a community or village level. And I'm happy to say that we are in the final stage of setting up an NDA at Nanikai, the pilot village for the PPOA. The second approach is coral restoration. Coral restoration program has only been done on one village, Antara, which is Nanikai, which is done last year with the help of a SPREP consultant, Mr. Carl. And the main goal of this program is to increase an ecosystem resilience to ocean acidification. The third approach is seagrass restoration. And seagrass restoration is also part of the SPREP PPOA project. It is also focuses on enhancing ecosystem resilience to ocean acidification. This was also done only in Nanikai village, Antara. The fourth is mangrove restoration. It is also part of the SPREP PPOA project, with its goal to help pH. However, mangrove planting is also done by the Ministry of Environment, and they have successfully planted mangrove on South Tara and some part of the outer islands. But we are happy that we can learn a few techniques from SPREP consultant, Dr. Po, on how to improve the survival rates of mangrove by growing them first in a nursery before planting them. That is why the picture shown shows mangrove which have already have leaves on them. Our vision is to extend these restoration approaches to the outer highland and to collaborate with regional bodies such as SPREP and SPC, and to collaborate with other stakeholders within Kiribati, such as Ministry of Environment, Land and Agriculture Development. Thank you. If you have any questions, then you can ask.
Thank you very much, Manibua. All right, so at this time, we're going to start the question and answer section. All right, so we got a ton of questions, and the way that this will work is I will read questions, and I think most of them are directed towards specific scientists. So when I call your name, you could unmute yourself, panelists, and respond to the questions. So this first question is for Duncan, and it comes from Chris Sabine. And he's asking, does SPREP have any formal connection to Hawaii? And how have COVID-19 restrictions impacted progress on the SPREP OA work?
Aloha, Chris. Thanks for the question. As far as formal connections with Hawaii, being part of the United States, the U.S. is one of our 5 metropolitan members. So the U.S. is a SPREP member. And as for COVID travel restrictions affecting our work, while we are trying to go ahead using local expertise on the ground in the countries where that's possible, and we have done quite well with that in some cases. For example, our work in Fiji is mostly able to go ahead without having to cross any international borders because our team there is based there. Some of the work has been necessarily postponed because we can't travel. For example, the coral restoration work in Tokelau. We started laying our transects and did our first assessments of the reef there, but we had planned to go back late 2019 to the 3 atolls of Tokelau to actually start the fragging and the coral planting, and that is now delayed until we can get back up to Tokelau. In Kiribati, as Manibua and Max so very nicely described, we were fortunate to complete most of the work there. I believe all of the work before the travel restrictions hit us, so did not affect our work in Kiribati. As for the work I described with KIOS, the Korean partnership to install MapCO2 OA monitoring buoys, we are on hold until our Korean colleagues can get back into Samoa to work with us to install that buoy. So the buoy arrived here, but we're not able to install it until we can get our full team in country. So some things are necessarily on hold, and where we can move things online for meetings and workshops like this one, we're doing that, and where we can rely on local expertise, we're doing that. Thanks, Chris.
All right, Duncan, I'm going to ask you one more question. This one is coming from Elizabeth Holland. The question is, are there any OA measurements being done in conjunction with the adaptation activities to document their effectiveness?
Yes, Bulabeth. We are in both Kiribati and Tokelau taking monitoring measurements of ocean chemistry in the sites where the adaptation is taking place. So in Tokelau, that's all 3 atolls. They have 3 ISAMIs currently in the water, recording probably the first ever time series of ocean chemistry in Tokelau. In Kiribati, that's another one I forgot to mention that's been delayed due to COVID travel restrictions. We have purchased an ISAMI to deliver to Kiribati, but it's currently waiting travel restrictions to allow us to do that. And so that will go into the LMMA that Manibua described in Nanikai Village. And as far as Tokelau, I did mention that we have a PhD student, Mildred from Papua New Guinea, who's studying in Australia right now. And as part of her research, she is going to make use of the time series, the OA time series from both Kiribati and Tokelau to try to investigate some questions as far as what effects coral restoration has in terms of enhancing reef resilience to OA. So those time series will help a lot with that research. Thanks, Beth.
All right, so this next question is for Antoine, and it also comes from Chris Sabine. Antoine, Suva is a very urban lagoon. Do you have a thought on the potential human contributions to the variability in that area?
Yes, thank you, Chris, for the question. Well, that is exactly what we are trying to measure with this project, is the impact of anthropogenetic inputs on the pH variability. So that will answer the questions on how much human activity is actually impacting the productivity of the lagoon, fisheries, and how that will, of course, impact food security and livelihoods of communities that depend on Suva Lagoon. So yes, it's very much what we're looking for. Thank you very much, Chris.
Okay, this next question is for Kim, and it also comes from Chris. It looks like there's more seasonal variability in the lower latitudes than in the higher latitudes. Was that expected? This seems counter to an expected temperature effect.
Aloha, Chris. Nice to hear from you. There's different drivers at the different sites. So temperature is a driver at one group of sites, but that's spread throughout the country. On the southwest of New Zealand, it's very high rainfall and high terrestrial runoff. So the variability at one of those southern sites is really driven by freshwater input. So both the DIC and the alkalinity are highly correlated with salinity. And the other sites, I think that really sort of shows the limitations of having a chemistry only type of program, is that we don't have a lot of auxiliary data to answer some of the questions around the drivers. So we have to make do with things like remote sensing and other programs that we can piggyback off nearby data. But we've been able to separate out primarily the temperature, the fresh water, and the biological drivers at the different sites. But to me, that really highlights that chemistry only is interesting, but it's not enough to really tease out exactly what's going on.
Thanks, Kim. This next question is for Duncan, and it comes from Denze. It says, thank you, Dr. Duncan. I would like to ask, how more can your projects impact directly the local communities in terms of uplifting their livelihoods? How do you conduct follow-ups on the awareness of the community to ocean acidification? What methods do you use to identify differences, changes, or purification from the acidified environments to a healthy ocean? and which method is used to eradicate alien or invasive species?
Okay, thanks for the question. Just try to think through the different questions one at a time. As far as raising awareness and trying to measure that, we usually, when we hold workshops or trainings, we usually ask participants to take a short survey pre-intervention and post-intervention to kind of gauge their basic knowledge of OA, its impacts, its drivers, and on the whole, when we have held interventions in our pilot site countries, we do show that participants report having learned or increased their awareness, their understanding of ocean acidification. So we usually show positive results with those types of pre and post surveys. The other question was, remind me the second part of the question.
The other question was methods that you use to identify differences or changes from an acidified environment to a healthy ocean, and then a last question about eradicating alien or invasive species.
Okay, so as far as monitoring the ecosystem health, yes, that is incorporated as part of the adaptation strategy. So at each of our pilot sites where we're doing an adaptation intervention, we have different monitoring protocols in place. So for example, when we do coral reef restoration, we have taken baseline surveys of several specific indicators of coral reef ecosystem health, and those might include percent ground cover, they might include invertebrates, they might include coral species diversity or fish biomass, for example. So we do baseline survey, and then we will continually, on an ongoing basis, our in-country partners have been trained on how to continue the monitoring. So obviously, after doing an intervention, the goal is to show some increase in a marker or an indicator of ecosystem health. So yeah, the monitoring and evaluation is a real important component. As for invasive species, they are absolutely a critical component of ecosystem health. It's not a specific mandate of the PPOA project to address that. SPREP has an entire program dedicated to invasive species eradication. So I won't try to speak for the people who know better about that type of work. We do collaborate, and we do, our partners in country wear many hats. For example, Max and Manibua and Kiribati from the Ministry of Fisheries, I know that they do work that addresses invasive species. So when they put together their LMMA, locally managed marine area management plan, they did include specific actions to address that, but the PPOA were not eradicating or directly addressing the eradication of invasive species. Thanks.
Thank you. This next question is for Patilla, and it comes from Max Rintal. Patilla, do you have pressure data for the time periods that you were taking measurements? Do the changes in pH and temperature variability correspond with the spring neap tide cycle?
Thank you. Unfortunately, we do not have the pressure data. We try to deploy our CTD, but we are still having issues with it. So we'll try to see how we can make sense of the data so we can tell a story. Thank you.
Patilla, there's also another question that came from Chris Sabine. Is it possible to get the local communities to sample for you if you cannot travel so far for your research site?
It's possible, but we have to find reliable people and train them. At the same time, there are health and safety issues associated with the sampling because we had to use mercury. So I can say it's not impossible. Thank you.
This is a question for both Duncan and Krishna. Can you please describe the role of the stakeholder workshops and how that will support SDG 14 reporting on OA? This is from Elizabeth Holland.
Krishna, you want to take it first? If not, I'm happy to go first. The role of the stakeholder workshops. Okay, so the stakeholder workshops were mostly, when it comes to the PPOA project, for education and awareness raising, training, and presenting and determining stakeholder priorities, presenting options for and determining stakeholder priorities for adaptation actions that the communities wanted to take. That was the main function of multiple iterative engagement workshops with the stakeholders in the communities. As far as the reporting against the SDG indicator 14.3.1, the pH indicator, that was supported by the monitoring efforts where we've partnered with the Ocean Foundation and GOA-ON to provide the GOA-ON in the box kits, and we've held several trainings. Several have involved Chris Sabin and at University of Hawaii. One was up at his lab. We've held trainings in Fiji, and those trainings were what directly increased the PI-TOA members and the GOA-ON in the box kit recipients' capacity to monitor OA and report it to GOA-ON and to the IAEA's OA ICC Coordination Center for collecting and collating that data. And IAEA as well has held some trainings where they've invited some PI-TOA members up to Monaco specifically for the management of that pH data for reporting against the indicator. Thanks.
Krishna, do you want to take that question? You could describe the role of stakeholder workshops and how that will support SDG 14 reporting. Krishna might have walked away from his computer for the moment. So let's take another question. This one is for Manibua from Chris Sabine. Are you able to make any OA measurements to document how your restoration efforts are impacting OA? Manibua, if you want to go ahead and unmute yourself.
If Manibua's unable to unmute, this is Duncan. I can mention that we have an ISAMI for Manibua's team there in Kiribati, but it's currently here in Samoa, and it would be there with them by now if it weren't for COVID travel restrictions. So one of the crippling things besides just being unable to travel is that mail itself in many cases is not able to travel. So we have their ISAMI here in Samoa, and that may be the answer to their ability to monitor OA at the moment.
Well, kind of along those lines, we have a question from Alexis from the Ocean Foundation. And this is actually a question for all of the researchers. Many of you expressed challenges with maintenance and troubleshooting the ISAMI sensor. What sort of support can GOA-ON or the Ocean Foundation provide to best help? Live troubleshooting sessions on a regular basis, or more manuals, or perhaps troubleshooting guides? Any one of the researchers can unmute themselves and answer.
This is Antoine. I'm speaking perhaps on behalf of us here at USP. So I think that we would need troubleshooting sessions, maybe online, and also first groups for treatments for internal and also pumps or whatever fails, so that we can always have some spares and know how to install them. That perhaps may help and also avoid some of the situations that Krishna and also us, I think, Katie, and we experience here.
I'm Patilla here. I also support live troubleshooting online for the Ocean Foundation to guide us through the process. I think there are already some very good guides, but I think it's still more clear if we have that live session guiding us in the maintenance and fixing the issues that we have. Thank you.
Hi, Alexis. This is Duncan. It might, you know, we have that resource, the OA info exchange, and we do, when I have troubleshooting, I do try to share it with everybody on there. But there may be, I don't know, another medium where we could keep a central repository of all the issues that people, not only in the Pacific, but around the world have had with an ISAMI. Usually I just go right to Sunburst Sensors and work over email with those guys to try to troubleshoot. But it seems like we should have a central repository, spreadsheet, whatever, Google Sheet, whatever it is, where we can list every time somebody's had an issue, what they've done about it, what the solution was, something like that, where we can all reference.
Hello, are you able to hear me? Krishna here.
Yes, we could hear you, Krishna.
Yeah, it seems that the organizer has muted until now. I have been requested. Okay, that's fine. In regards to the troubleshoot, as I submitted in my presentation, we should have a regional hub where we can have these materials or some spare parts being available. So it will be quick for us to exchange or to hire or loan, something like that, rather than every time reaching to these quotations from their funding and all these kinds of things. And as Antoine said, that it would be fine for us to have some sessions within ourselves, especially the Pacific Islanders researchers who are doing this OEM monitoring. That would be very good to exchange thoughts. Thank you. Hello. Hello.
Yes, we could still hear you, Krishna. All right. None of the other researchers have anything to add. We could take a couple more questions before we end this session. All right. We have a question from Roshan Ramaswar. This is a question for both Patilla and Krishna. The question is, ISAMI pH sensor deployment can be updated. Have you made any quality controls, measurements, or adjustments?
Thank you, Michael. Patilla here. I have not yet gotten up to that stage. As I said before during my presentation, I'm currently processing the data so that we can look at how to do that. Thank you.
Krishna, do you have anything to add?
Yes, as I told you in the presentation, Roshan, we have collected some data and we will surely pass it over to the Sunburst and see whether there is a quality control is required, and also share with the other Pacific Islanders. And as part of the updatement of the firmware, they have directed us, and we have done that, and we could have some clear data. But unfortunately, the whole sensor is currently not working, and we are looking at how we can have a replacement of that. Thank you.
All right, I want to thank all of our attendees and all of our panelists today for sharing those questions and for presenting those wonderful talks and answering all those questions. Before we leave today, I want to mention that we want to continue the conversation on Twitter. So continue using the hashtag OA Week 2020 and please pose more of your questions to these speakers on that platform. You also could use the Ocean Acidification Information Exchange. A lot of these researchers also have presences on that platform. And before we go, I just want to mention that we have a few other sessions happening today. The next session will be the OA Mediterranean Hub that will be happening at 12 noon Central European Summertime. And the last session of day one will be the North American Hub at 12 noon Pacific Daylight Time. Thank you all for attending the very first session of OA Week 2020. And I hope to see you all at the next 9 sessions. Have a wonderful day. Bye, everyone.